Welcome, science friends, to mitosis, also known as M phase. M for mitosis. So there's a few different stages of mitosis. We start with prophase, move on to metaphase, anaphase, and finally telophase. Prophase is the first stage of mitosis. Few different important things happening here. So first, the centrioles or centrosomes start to separate from each other. So when they're hanging out in the cell before mitosis, they're just kind of together chilling. But they have a job to do here because they're the ones running the show for cellular division. So the centrioles need to separate from each other and spindle fibers begin to form. So in our drawing here, you see those little like arm, leg, noodly things. Those are spindle fibers, and they're going to have a pretty important job in the next couple of steps. Next, the chromatin, so the noodly DNA stuff hanging out inside the nucleus, that condenses into our chromosomes. So we replicated our DNA, and now we need to make sure all of our information is paired up correctly and it gets to the right place. We don't want duplicates in our new cells. We want each cell to have all of the stuff it's supposed to have. And then finally, the nuclear envelope, so that membrane around the nucleus, starts to dissolve so we can get to those chromosomes for our cell division. The next step is metaphase. So spindle fibers, so now they're much longer. You can see that the centromeres have moved to opposite sides of the cell. Those spindle fibers are going to attach to the chromosome centromeres, so the little buttons that's holding those X shapes together. And those chromosomes start to line up, and they're lining up along the middle of the cell, and that has a special name called the metaphase plate. So really a lot of movement happening in metaphase. So we see they're lined up, the spindle fibers are attached to the centromeres, and we're ready to go to the next stage, which is kind of fun and crazy. Here is an example of sort of prophase and the beginning of metaphase. We're seeing those centrioles move to opposite sides of the cell, their spindle fibers are extending, those chromosomes were released, they're condensing into chromosomes, the chromatin, then the nuclear envelope dissolves, and they're starting to move towards the center of the cell and attach to the spindle fibers. The third step is called anaphase. Anaphase, in my opinion, is like one of the most exciting phases. Those X shapes, those chromosomes, are going to pop apart into their sister chromatids. Now, we went through the DNA replication where we had copied all of that genetic information to those two different parts of our X-shaped chromosome. And so now we popped them apart, those spindle fibers went boop, and now one chromatid from each of the chromosomes is going to the opposite side. So those spindle fibers are starting to pull them apart towards the opposite sides of the cell. We can see that here in this animation. We just boink, popped open that X shape, and now they're going to opposite sides of the cell towards their centriole. The last stage of mitosis is called telophase. This is sort of like a relaxing of the process. Okay, we did it, we separated all the chromosomes, now we just need to kind of take it easy. The chromosomes that were moved to opposite sides, they start to unwind back into their chromatin from the chromosome, so from the X to the noodly shape. Our nuclear envelope or membrane is starting to reform around them to keep that DNA safe. And we're starting to see a sort of beep, a little pinch. It's going to become two new cell membranes for each of our daughter cells. So it's not quite popped apart just yet, but we're starting to see, okay, it's almost getting a waist. And then the last part here, it's technically not part of mitosis, but it's kind of finishing up the process. It's called cytokinesis, or sometimes referred to as C phase, C for cytokinesis. And that's just the actual physical separation of these two new daughter cells. They should be identical to each other because they all have the same copies of DNA that were pulled apart during the anaphase process. And they're brand new, happy, ready to go cells to do their cell jobs. Here's an animation to show us both telophase and cytokinesis. So in telophase here, we see, okay, we're putting the nucleus back together, we're unwinding our chromosomes back into chromatin, and we're separating during we see a little bit of a pinch for telophase, and we are separating for cytokinesis. It's really important to remember the steps and the order of this mitosis process. We're not really going to talk about interphase a lot in this video, but that'll be in our next video. But there's a few different mnemonics or memory devices that people can use. I personally like pass me another taco. So P for prophase, M for metaphase, A for anaphase, T for telophase. It's just another way to remember the order because I love tacos. If you don't know already, I love tacos. So it's just a cute little mnemonic to be, okay, pass me another taco because I always want tacos, and it's in that order. If you want to include interphase, you can just add the I and make up a new mnemonic device. I picked my apples today. Again, I for interphase, P for prophase, M for metaphase, A for anaphase, T for telophase. Here's another animation to sort of show us all of the different processes here, going in order. And so here we start at interphase. We start going through prophase, metaphase, anaphase, telekinesis, and cytokinesis. Again, this one's a much lower resolution, kind of an 8-bit animation of the mitosis process. That was mitosis or M phase. I hope it helped.